everyone. Hope you're doing well. I'm re-recording my final review for a couple of reasons. One is because I had myself on mute during the review session, and then second is all my Q&A material was deleted right before the review session, and so that was my fault. And what I have here is an extended version of the midterm flowchart, which I'll be sending out. Definitely go check out my midterm review video. You can find that on my channel for a more comprehensive explanation of everything in the first half of the class. But I'll be highlighting some key points here that you should know for the final. And you can reference that midterm review if you want further explanation. Okay, just a couple things about the final though. It'll be 32 questions and a mix of free response and multiple choice. I honestly think the final is a bit better than the midterm. And yeah, hopefully it shouldn't be too much of a problem with time constraints or anything like that. Open note, everything. Also, feel free to submit the report tonight. I don't know why the deadline was 5 p.m., but I'll fix that. Anyway, okay. So the way we started the class was looking at Bacillus subtilis, which had this alpha amylase gene, which we're then cloning into this vector plasmid into E. coli, and then transforming into E. coli DH5 alpha and then into BL21 for expression. So let's start over here with our genomic DNA extraction. So we extracted all of the DNA in the Bacillus subtilis organism and measured that, looked at the quantity, purity, and quality of the DNA. And then here's where we have to take a look deeper is PCR amplification. Okay. So with PCR amplification, we are trying to amplify our gene of interest among all the genes, all the DNA in Bacillus subtilis. So that includes 5 steps here: our full denaturation, our denaturation annealing extension, which goes on for 30 cycles, and then full extension at the end. Okay. So one of my questions I want to ask you guys is, I was going to ask which of these steps is primer dependent. I have it right here, but why? Why do you think that the annealing, also known as the melting step, is primer dependent? So you can pause and think about that. But really it's because the annealing temperature refers to the temperature at which the primers will re-anneal onto the target strand of DNA, right? So this temperature is very important. If it's too high and too low, it'll have different consequences. So I want you to pause and think about what would happen if the melting temperature or the annealing temperature of the PCR program was too high compared to the actual melting temperature of the primers. Okay. And then think about if it was too low, if it was too cold. Okay. So when you're ready, if the temperature was too high, that means that the primers never got a chance to re-anneal. It would never reach a stable enough condition that those primers would re-anneal onto the target strand, right? And that, if you translate it onto a gel, would mean no bands, right? Because we're not able to amplify our gene anymore, any gene. If the temperature was too low, this means that the primers could potentially be too stable at places they shouldn't be, and potentially non-specifically amplify random genes that were off target, right? So that would lead on a gel, that would lead to multiple bands, okay? Multiple bands. What we're really looking for is just one band, because we're looking for a single amplification of a gene, okay? Our gene of interest. All right. So other things we can take a look at here are the primers themselves, right? So we need a forward primer, we need a reverse primer, we also need our target DNA, we need Taq polymerase and buffer, as well as cofactors like magnesium. One thing I'm missing here, I want you to pause and try to guess what it is. I'll let you take a second. We have our Taq polymerase, our target strand, forward primer, reverse primer, all the equipment. But we're missing our building blocks. We're missing our DNA or our nucleotides to make more DNA, right? So that's very important. So we need all those nucleotides. Okay. Now, how do we get primers? Well, the pragmatic way would just to use a program online. There's many programs that you can use, but if you're not comfortable with those, it's honestly just easier to do it by hand for the exam. For forward primers, I want you to pause and think about what would a forward primer look like. How do I make a forward primer if I have the template strands? Okay, and then pause and think about how would I be able to get the reverse strand? How is that different? Okay, so here's a hint over here how I've drawn this out. If we're looking for with the forward primer, there's no need to make a complement. There's no need to invert or reverse the orientation. Simply just take the first however many you need base pairs, let's say 20 base pairs, and that will actually be your forward primer. Just take your template strand that you're given, 5' to 3', and that is your forward primer. For reverse primer, it's a little bit, there's a couple steps. So for reverse primer here, the first step would be to, let's say 20, to get the last 20 base pairs, find the complement of those, okay? And then at that point, you're looking 3' to 5'. The last step, when you're inputting your answer, is to invert that, right? To turn it to 5' to 3', because that's always how we input our DNA sequences, right? So forward primer would just be straight up the first 20 base pairs that we see. Reverse primer would be finding the complement and then inverting that, or just reorienting it, the sequence, so it's 5' to 3' instead, okay? All right, so that's all for PCR. So we have our amplified inserts that we're ready to insert into our plasmid. Where did our plasmid come from? So let's see over here. So here we have extracted our plasmid through plasmid extraction, and this plasmid contains some very important components. We have our SAC-B, which is our counter selector gene. Okay, we'll be talking more about that. Our ampicillin resistance, that's our selection gene. We have our origin of replication. We have our LAC-I repressor. That's going to help us control expression. We'll talk more about that. We have our T7 promoter, which is recognized by the T7 RNA polymerase, which will go ahead and express our gene. This region I'm highlighting here is exactly the multiple cloning site, exactly where our gene will be inserted. Okay, and that occurs using restriction enzymes. And in our class, we use ligase independent cloning. Okay, so in this multiple cloning site, there's also multiple, there's more components here. We have our T7 promoter, which is right here. We have our his tag, our histidine tag, which is a string of about, let's say, 6 histidines. That'll allow us to select for our protein during purification. We'll talk about that later. We have our LAC operator, where our LAC-I repressor is going to bind right on our promoter region. And then we have our restriction sites, which are around. We have thrombin, okay? I want you to think about what thrombin does. If you can, pause and think about what thrombin does. Thrombin is a protease which will cleave the site that it's attracted to. So here there's a thrombin site, okay? And the reason we want a cleavage site there is because we have our his tag, right? And that's great for purifying, but after, we want to make sure that when we're testing out our protein, it doesn't have extra unnatural components that weren't supposed to be there. So the last thing that you can do before testing out your protein, after you've already purified it, you got it, is you can just cut off that his tag using thrombin protein. Okay, and then we also have our T7 terminator. Okay. So another reminder here is that DNA appears, if you were to load a plasmid on a gel, it would most likely appear in 3 forms. Hold on real quick, I have to charge my laptop. Okay, so plasmids are found in 3 forms, the supercoiled, linear, and circularized. So those will appear differently because they're different shapes on the gel, just a reminder there. Okay, so once we have our plasmid, we need to do a restriction digest because we want to remove that SACB gene, right? That is lethal to the cell. It's a suicide gene. And if the plasmid still contains it and is inserted into E. coli, then it will kill the cell and also denote to us that our insert has not been inserted. Okay, so we are using restriction enzymes to cut at the restriction sites, cut out this SACB. Okay, and a question here is, if we want to ensure that our insert, our gene of interest, our target gene, is inserted in the right direction, then how do we do that? And here I have the answer. Is you use 2 different types of restriction enzymes. Okay, so if you have 2 different types of restriction enzymes with different sticky ends, then there's no way that the gene can insert in the wrong direction. Okay, if you have 2 of the same restriction sites, then it's possible it could be flipped. Okay, all right, so please be familiar with restriction enzyme analysis. For example, over here on the right, just be comfortable with cutting plasmids and then visualizing that on a gel, right? Okay. So you can also refer to my review about the primers, but basically you can use different primers in different locations, as well as restriction enzymes to analyze if your insert has been inserted properly after the fact. So that's just by taking advantage of the different positions of the primers, especially one that's an internal primer within the gene, right? And that'll provide different lengths. Okay, if the gene wasn't inserted there, you might not get some bands, or they might be different lengths. Okay. All right, so if you visualize that on a gel, after you've cut out SACB, it should look like this. We have our linearized vector plasmid, and we have SACB cut out over here. Okay, now once we do our ligase independent cloning, put our gene inside our plasmid, then we transform that into DH5 alpha. Okay, one thing I would want to really emphasize is, so we do our heat shock transformation, right? And get those plasmids in our DH5 alpha strain. Our DH5 alpha strain is best for, it's known as like a cloning vector. It's good for as a cloning bacterial strain. It's extremely crucial, and I honestly cannot highlight this enough. Please don't forget this component here, okay? We need to grow after the plasmid has been inserted into a bacteria. We have not given them a chance to express the plasmid genes yet properly, okay? So I want you to think about why. Think about why the cells would immediately die, okay, if we plated them on this type of agar, okay? So if we plated them on sucrose and ampicillin, Well, theoretically, sucrose should only kill the SACB, the organisms that still contain the SACB gene, right? But actually, if you plate them initially, if you plate them before growing them for an hour, for incubating them for an hour on super optimal broth, they're going to die because of the ampicillin first, okay? Because the sucrose, it takes a minute for it to, you know, to ramp up the sucrose, the lethal sucrose pathway. However, the ampicillin will kill the cell right away because it has not had a chance to develop ampicillin resistance using the ampicillin resistance gene, right? So that is super crucial, okay? That's really important to keep in mind. Okay, we did colony PCR, and then we transformed into BL21. This is an expression vector, and the reason it's only used as an expression vector is because it has the T7 RNA polymerase gene in the genome already ready to use, okay? And it already has a regulatory system for it. And it's actually identical to the regulatory system we have for our target gene using that LAC-I repressor. However, there are other strains and the inducer can change depending on the type of strain that there is. So typically, we're going to be using IPTG for the induction of the T7 gene and our target gene. So let me clarify this here, okay? So IPTG, again, it's a synthetic mimic of allolactose, okay? Allolactose binds to the LAC-I repressor and stops it from repressing. And it inhibits it from repressing any longer, okay? So IPTG typically acts on the T7 polymerase gene as well as our target gene, okay? On our plasmid, we have the LAC operon or operator is what I really should have put here. LAC operator. My bad. Okay. The LAC operator is going to be somewhere around there. Okay. So the IPTG induces in two separate things. Okay. First, it induces the T7 polymerase gene to form or to transcribe and translate the T7 polymerase itself, because we need the T7 polymerase to then go ahead and read our target gene. Okay. So we have IPTG, which we add when the OD is 0.5, when it's in exponential phase. Okay. We had our IPTG in blue here. Okay. That tells our LAC-I it's time to go. And then the T7 polymerase can be transcribed, translated. We have T7 polymerase now. Okay. That's going to go ahead and read our target gene. But again, we have LAC-I here. However, IPTG binds allosterically to our LAC-I, telling it to move. And then our T7 polymerase can read the T7 promoter, which is over here, and then read that target gene, okay? In our case, alpha amylase. So it's basically what's going on. But again, this repressor here doesn't have to be LAC-I, and the inducer doesn't have to be IPTG. It could be other things, like in the practice questions. Okay, think about the order that this needs to happen, right? So I went step 1 to step 2 here. Okay, think about that logically, and why it goes in this sequence. Okay. So we also have affinity purification. We use nickel affinity chromatography. And the first step here is to bind our protein to our column, right? That has nickel in it in this white segment here, right? So, sorry, in red, we have our target protein and it has a his tag. So this protein here in red has this his tag. Think about where we want this his tag located, okay? We don't want our his tag in an inaccessible place. We want it as clear and obviously out of the protein as possible. This doesn't mean that it's necessarily in the front or the back of the protein. You can put the his tag on the N terminus or the C terminus, wherever is better. If neither work, maybe it's time to get a new type of tag. But as long as it's accessible, it should work. And we make sure that it's 6 histidines long, not something like 1 or 2, because the binding affinity will not be strong at all, number 1. Number 2, many proteins have histidines in a row, right? So we want something that's unnatural, that's not going to happen randomly, and that will create a strong bond with the nickel in the column, right? Okay. So now we have our red protein bound here. We also have garbage, other types of protein in the cell-free extract that we added, right? We're going to wash that out. Hopefully only our protein should bind, our protein of interest. And then the last step would be to elute our red protein of interest out, okay? So this can happen in 3 different ways. One is using competition with imidazole, which competes with the nickel bonds and then leaves the protein unable to match those or to find bonds. We also could use pH change to deprotonate the histidines. So there's no longer that affinity for the nickel because it's dependent on the hydrogens on the histidines, I believe. And then a last technique could be stripping the nickel and histidines together, okay? So this means basically cutting off the nickel with the protein and you end up with protein and nickel together, which you can remove later. But yeah, so that's with EDTA, okay? So now once we have our purified singular protein, then we can characterize it. Okay, so we can characterize it using SDS page. We can look at the size and we can also look at the relative quantity. If you have different tubes with different concentrations of the same protein, let's say, and you want to see, which tubes have the most concentrated proteins, you can see that by the thickness of the band in the gel, right? So if you have more protein, the thicker the band's going to be. Okay, so importantly, SDS page is a denaturing gel. Okay, this means that we're going to destroy the functional properties of this protein, and it's going to involve this lamellae buffer. Okay, this includes SDS, which is also present in the buffer used to run the gel. But this SDS creates a charge uniformity, right? Because proteins, because they're various amino acids, have different charges, like, positive and negative charges, not necessarily uniform or a net negative, okay? So that's important to do because we want to drive this protein down. So if you notice over here, we have positive and negative charges, and we want all the charges to be negative here. We can do that with SDS. It will drag the protein down to, I want you to pause and think, the anode or the cathode. And the answer is the cathode, toward positive charge, just like in gel electrophoresis. Additionally, having proteins denatured by beta-mercaptoethanol or DTT denatures it further. The disulfide bonds are the hardest to break, and that's what it works on, and it activates by boiling. It also denatures it further when you boil. Having a protein denatured is really important to be able to fairly compare the size between different proteins as well as against the marker, right? We want to be able to compare to the latter properly, right? So if we did not denature, then we would end up with proteins of the same size, but still at different heights and different heights in the gel, basically. Okay. So standardizing the shape and the charge is very important. Okay. Then this is an example we went over today, which is basically, you know, if you have the marker here, what would the self-free extract look like on a gel? That would just look like a ton of different proteins at different sizes. And here you have before induction, after induction, and this light blue is your protein of interest. It should be a nice thick band in there. And then once you purify, it should also be a nice thick band. Think about how we know if our protein was induced or not, right? So, or if our protein was properly expressed. You can analyze that by looking at the self-free extract before induction and after induction. If this blue band here wasn't here, if it wasn't present, then that could tell you that the protein was never expressed, right? And you would see the same, you would see no band in the elution. This is the elution. The elution lane. Okay. So alongside the SDS gels, we also have native gels or zymograms. That allows us to test the function and potentially measure the charge. We didn't do that in this class, but the native gels are able to separate by charge because they don't uniform the charge. They don't denature the proteins as well. And that's really important to be able to still test the function of the protein, right? So we looked at the zymograms with iodine and starch. We got to see our protein alpha amylase degrading the starch in the gel itself, in the matrix itself. So that's another important component. Okay. We also looked at starch plates. Well, briefly, we talked about it for like a millisecond, actually. Starch plates are like blue plates that if you smear bacteria on there, then they will start to hydrolyze the starch, the blue colored starch around it, forming a clear halo if it contains genes like alpha amylase or other genes that break down starch, right? That's an important thing to remember. And then the starch iodine test, which we also looked at in class, where the iodine is like a ball. Okay, the iodine molecule is like a ball, and the starch, the amylose specifically, forms a helical structure where the iodine as a ball goes and fits inside within that helical structure. When alpha amylase breaks that amylose structure, because it goes around cutting the starch, then the iodine is no longer as dark as it should be, or as dark as it once was. And that allows you to test the ability. And that's exactly how the zymogram works as well. Okay. We also looked at the glucose determination steps. That, keep in mind, is a red color, right? So we looked at in the last class how well alpha amylase versus glucoamylase can turn starch into glucose. Okay, so in starch iodine test, we looked at how much starch was left over. Glucose determination, we looked at how much glucose was left over. Okay, here's kind of a good description of a simple description of what you should expect. With alpha amylase, it's like you have a big tree of amylose, of starch, okay? And you're cutting off big branches at a time. It's an endoglucanase. Whereas glucoamylase is like going at the ends of each of the branches, the very tips, and taking off like a leaf at a time, a glucose molecule at a time. That's kind of how it works. Whereas if you combine the power of alpha amylase and glucoamylase, well, now at the same time, you have alpha amylase breaking down branches and glucoamylase shredding the ends as they're being cut down by alpha amylase, right? So that would be the most effective. Just to highlight here, alpha amylase doesn't really break starch down into glucose, mostly oligosaccharides, because they're endoglucanase, yeah, I guess. And then the glucoamylase is an exoglucanase. So it cuts off from the ends, from like those reducing sugar ends, right? And if you use the power of both of them, it'll be the most effective at producing glucose. And so you'll get a mixture of glucose and oligosaccharides, but definitely more glucose than the glucoamylase, right? Okay. So I think I pretty much covered everything that we talked about. Yeah. If you guys have any more questions, please feel free to email me. I'm happy to go over any of the practice questions with you guys or anything in general. Email me whenever. I'll try to get back to you as soon as I can. Yeah, I hope you found this somewhat helpful, and I hope you guys do well on the exam. I'm really confident you guys will, and I'm really proud of you, and I had the best time ever teaching. So thank you. See you soon. Bye.